Hello students, welcome to EPG Pathshala. I am Dr. Switi Shrimali from the Department of Zoology in Acharya Narendra Dev College, University of Delhi. In this lecture, I will be dealing with a module from immunology. The title of the module is Antigen Processing and Presentation to T lymphocytes, Antigen Presenting Cells, and Role of CD1 in Antigen Presentation. So after going through this module, you should be able to differentiate between the modes of antigen recognition by B and T lymphocytes, understand the concept of self-MHC restriction, appreciate the need for antigen presentation to T lymphocytes, understand how the exogenous antigens are processed and presented, describe the processing and presentation of endogenous antigens, understand the role of antigen presenting cells, appreciate the concept of cross presentation, and understand how non-peptide antigens are processed and presented. Before we go any further, let us first discuss the meaning of antigen processing and presentation. An antigen, as you all must be aware, is a molecule that is capable of binding to a specific receptor of the immune system. These receptors are encoded by B or T lymphocyte. The B cell receptor and its secreted form antibody recognize their antigen in their natural 3D conformation. Further, antigen recognition by BCR occurs directly, irrespective of whether the antigen is present on the surface of another cell or is drifting independently in the bloodstream or interstitial spaces. On the other hand, T cell receptor can recognize an antigen only after it has been first broken down into small peptides by a process known as antigen processing, and secondly, if it's presented in the context of a second MHC molecule, self-MHC molecule. Thus, antigens drifting independently in the bloodstream or interstitial tissue spaces are never directly recognized by the T cell receptors. Such antigens are only recognized by the TCR after being processed and presented by an appropriate cell. So how did we come to know that antigen processing is essential for T cell activation? Ziegler and Unanue in 1981 and Shimonkevich in 1983 proved beyond any doubt that antigen processing is essential for activation of helper T cells. In a nutshell, these experiments showed that, first, when an APC presents an antigen to a T helper cell, the T helper cell with the cognate TCR gets activated, which is usually what happens. If, however, the antigen processing cell is fixed and then an antigen is provided, the T helper cell with the cognate TCR does not get activated. On the other hand, if the APC is provided with antigen 1 hour before it is fixed, then the APC is still capable of activating the T helper cell with the cognate TCR. Interestingly, even a fixed APC can activate the T helper cell if the APC is provided with an antigen which has been pre-digested into peptides. So together, these experiments proved that antigen processing is required for the activation of T helper lymphocytes. The proof for the requirement of antigen processing for the activation of cytotoxic T lymphocytes came from experiments by Townsend. So these experiments proved that antigen processing is a prerequisite for activation of both helper and cytotoxic T lymphocytes. So let's ask ourselves another question, and that question is, how do we come to know that T cell activation will only occur when the antigen is presented in the context of a self-MHC molecule? In 1973, Rosenthal and Shivach provided experimental proof for self-MHC restriction of T helper cells. So how did they do it? They isolated peritoneal macrophages from a guinea pig, say guinea pig 1, and then incubated these with the antigen. Meanwhile, the same antigen was introduced into another guinea pig, say guinea pig 2, and after a week, antigen primed T cells were isolated. These antigen primed T cells were then mixed with macrophages from guinea pig 1. T cell proliferation was estimated by quantifying DNA replication by measuring the incorporation of tritiated thymidine. As you can see in the figure, the presence or absence of T cell proliferation depended on the haplotype of the two guinea pigs. T cell proliferation was observed only when the T cell and macrophage had the same haplotype, thereby demonstrating self-MHC restriction of helper T cells. So how about the self-MHC restriction in case of cytotoxic T cells? How was that proven? In 1975, Zinkernagel and Doherty provided evidence that self-MHC restriction occurs in case of cytotoxic T cells also. In their experiment, they used a CBA-H strain mouse with H2K haplotype and immunized it with the lymphocytic choriomeningitis virus, which is also known as LCMV in short. After some days, its spleen was extracted and a splenocyte suspension was prepared. This splenocyte preparation contained cytotoxic T cells specific for the LCMV antigens. Meanwhile, a fibroblast cell line was either incubated with the LCMV virus or was left uninfected. Both these sets of fibroblasts were grown in a culture medium containing a radioactive isotope of chromium, which is chromium-51. These LCMV infected and uninfected chromium-51 labeled target cells were then incubated with the cytotoxic T cells from the CBA-H strain mouse. The killing of these target cells was now estimated by measuring the release of labeled chromium into the culture supernatant. This assay is known as chromium release assay. You can see in the picture, this is what we are trying to show. The mouse is being immunized by LCMV virus and its spleen cells are getting extracted. And then on the right hand side, you can see the fibroblast cell line being labeled with chromium-51. And then both these kinds of cells are incubated together and the measurement of chromium-51 released into the culture supernatant is done. So what did they find? Only those target cells were killed which satisfied the following two conditions. First, they were infected with LCMV, which means that the uninfected target cells were not killed. This observation suggested that antigen presentation was indeed essential for cytotoxicity mediated by the TC cells. The second condition that was necessary for the killing was that they shared at least one class 1 MHC haplotype with mouse 1, which was the source of cytotoxic cells. This observation proved that cytotoxic T cells or TC cells also exhibit self-MHC restriction. Now the same experiment was repeated with mice having different haplotypes such as H2A and H2D and even congenic mice which acting as the source of cytotoxic T cells. All of these experiments provided the same results. So the inference from all these experiments was also the same, meaning that cytotoxic T cells exhibit self-MHC restriction. Now that we understand the importance of antigen processing and presentation for T cell activation, let us discuss how these processes are carried out. Depending on whether the antigen is translated inside or outside the cell which process and present it, there can be two types of antigens. Antigens translated within the cytoplasm of the cell that presents them are known as endogenous antigens, while those translated by the cells other than the cell that presents them are known as exogenous antigens. Now these endogenous and exogenous antigens are processed in different ways. Let us first consider the processing and presentation of the endogenous antigens. Endogenous proteins of the cell are presented in the context of MHC class 1 molecule. These are translated and assembled on RER membrane. RER stands for rough endoplasmic reticulum. As is apparent from the figure, endogenous proteins are broken into small peptides in a protein assembly called proteasome. Antigen is then transported into the rough endoplasmic reticulum via an ATP-dependent process mediated by the TAP protein. TAP stands for transporter associated with antigen processing. In the rough endoplasmic reticulum, Tapacin, which is a protein associated with TAP protein, recruits MHC class 1 molecule to TAP by binding to another protein known as ERP57. This complex of TAP, tapacin, calreticulin, and MHC class 1 molecule is known as peptide loading complex, or PLC in short. As its name suggests, PLC loads peptides on MHC class 1 molecule. Besides recruiting MHC class 1 molecule to TAP, tapacin also performs peptide editing, a process which involves exchange of low affinity peptides on MHC class 1 molecule with high affinity peptides. The ideal size of peptides for loading on MHC class 1 molecules is 8 to 9 amino acid. The PLC loads the right size peptides on MHC class 1 molecule straight away without exposing them to the ER lumen. However, longer peptides are trimmed from their amino terminus by the enzyme ER aminopeptidase 1, in short ERAP1. After the desired length is reached, the PLC loads the peptide on the MHC class 1 molecule. The binding of peptide to MHC class 1 molecule stabilizes it. The peptide MHC complex then gets dissociated from the PLC. From the RER, peptide MHC complex moves to the Golgi apparatus and is then presented to the cell surface. The peptide MHC complex may now be recognized by a cognate TCR on a memory cytotoxic T cell. In the presence of appropriate co-stimulatory signal, the memory T cytotoxic cell will get activated. However, the activation of naive T cells is usually brought about by a special type of antigen presenting cells known as dendritic cell, or DC in short. Let us now discuss the processing and presentation of exogenous antigens, which are those antigens that are not made inside the cells that present them on their surface. The exogenous antigens are presented in the context of MHC class 2 molecule, which are expressed by a special class of cells known as antigen presenting cells, or APCs. There are two kinds of APCs, professional and non-professional. Professional APCs express MHC class 2 molecules and co-stimulatory molecules, for example CD40 and B7. The examples of professional APCs include dendritic cells, or DCs, macrophages, and B cells. Non-professional antigen presenting cells, on the other hand, are those cells that can be induced to express class 2 MHC molecules and or co-stimulatory molecules under special conditions such as prolonged inflammation. Examples of non-professional antigen presenting cells include fibroblasts, glial cells, epithelial cells in thymus and thyroid, pancreatic beta cells, and some endothelial cells. The exogenous pathway of antigen presentation begins with the internalization or endocytosis of the antigen by the antigen presenting cells. The nature of the antigen determines the mechanism of their internalization into antigen presenting cells. Particulate antigens, which are bigger in size, are internalized by phagocytosis into organelles called phagosomes. Soluble antigens, on the other hand, are internalized by pinocytosis or receptor-mediated endocytosis into organelles called early endosomes. Once internalized, the phagosome and the early endosome undergo maturation. This maturation involves many sequential changes such as acidification of the lumen and acquisition of new proteins by fusing with organelles such as lysosomes or vesicles. Finally, the phagosome and the early endosome mature into a special type of late endosome called the MHC class 2 containing compartment, or M2C. In M2C, proteases called cathepsins degrade the invariant chain, leaving behind a peptide of 20 amino acids called the class 2 associated invariant chain peptide or CLIP. Antigen binds MHC class 2 molecule by replacing CLIP and then gets broken down into small peptides by cathepsins. The protein HLA-DM facilitates the loading of antigens by removing CLIP from the peptide binding groove. Further, it favors loading of high affinity antigens, a process which is known as peptide editing. Another protein, HLA-DO, inhibits this HLA-DM mediated exchange of CLIP with the antigen. Hence, HLA-DO and HLA-DM have a significant effect on the kind of antigens that get presented, and thus they are capable of modulating immune responses. Once the MHC and peptide complex is formed, it is presented on the surface of the antigen presenting cell where the helper T lymphocyte interacts with it through its T cell receptor. In the presence of appropriate co-stimulatory signals, the memory T helper cell can get activated by any of the antigen presenting cells. Unlike the memory T helper cells, the naive T helper cells are usually activated only by the dendritic cells. So we can see that it is quite clear that the exogenous and endogenous pathways of antigen presentation differ greatly in several key aspects. Let us now summarize these differences between the endogenous and exogenous pathway of antigen presentation. The first point of difference is that in case of endogenous pathway, the point of origin of antigen is internal. That is, the antigen is formed within the cell that presents it. Whereas, in case of the exogenous pathway, the point of origin of antigen is external. That is, the antigen is formed outside the cell that presents it. Another point of difference is that while all the nucleated cells are capable of presenting antigen via the endogenous pathway of antigen presentation, on the other hand, only specialized cells called antigen-presenting cells are capable of presenting antigen via the exogenous pathway. The third point of difference between the endogenous and exogenous pathway of antigen presentation is that while the endogenous pathway does not rely on phagocytosis or endocytosis for internalization of the antigen, the exogenous pathway relies completely on these two processes for internalization of the antigen. The fourth point of difference between these two pathways is that in the endogenous pathway, proteolysis is mediated by the proteasome complex, which is present in the cytoplasm. However, in the exogenous pathway, proteolysis is mediated by certain kind of proteases known as cathepsins. The fifth point of difference between the two pathways is that endogenous pathway usually presents peptides which are about 8 to 9 amino acids in length, while exogenous pathway presents peptides that are almost 14 to 20 amino acids long. Further, endogenous pathway involves antigen presentation by MHC class 1 molecule, while exogenous pathway involves antigen presentation by MHC class 2 molecule. Another point of difference is that the peptide loading complex or PLC, which comprises of proteins such as TAP, Tapasin, ERP57, calreticulin, etc., are involved in peptide loading in the endogenous pathway. On the other hand, the proteins involved in peptide loading in exogenous pathway include invariant chain, HLA-DO, HLA-DM, etc. The final point of difference between these two pathways is that the peptide MHC complex formed at the end of the endogenous pathway presents antigens to CD8 positive cells, which are basically cytotoxic T cells, while those formed at the end of the exogenous pathway presents antigen to CD4 positive cells, which are essentially helper T cells. So far, we have discussed only two different pathways of antigen presentation. Do these pathways ever merge? That is to say, is it possible for an exogenous antigen to be presented via machinery for endogenous pathway? Turns out that it is possible. So how does that happen? The answer lies in a process known as cross-presentation, which occurs in dendritic cells. Cross-presentation involves the uptake of exogenous antigens by phagocytosis or pinocytosis, followed by its presentation via the endogenous pathway with the MHC class I molecule. Now, two routes can explain the events involved in cross-presentation by dendritic cells. The first route involves the uptake and processing of the exogenous antigen in the late endosomes. Peptides are loaded on MHC class I molecules in the late endosome, and then the peptide MHC complex is transported to the surface of the dendritic cell. The second route also begins with the uptake of the antigen using the endocytic machinery. Subsequently, the antigen gets translocated out of the endosome into the cytosol. This is followed by proteasome-mediated proteolysis of the antigen. Peptides are then translocated into the rough endoplasmic reticulum by TAP protein. Peptide loading complex then loads the peptide on MHC class I molecule. The peptide MHC complex thus formed is then transported to the surface of the dendritic cell. If this peptide MHC complex is presented with appropriate co-stimulatory signals to a naive cytotoxic T cell, it gets activated to cytotoxic T lymphocyte. Now you may ask me, are all our dendritic cells capable of cross-presentation? The answer is no. Not all the dendritic cells are capable of cross-presentation. The process that enables the dendritic cells to perform cross-presentation is known as dendritic cell licensing. At first, the dendritic cell processes and presents an antigen with MHC class II molecule using exogenous pathway, thereby activating the helper T cell carrying the cognate T cell receptor. The activated helper T cell then provides the necessary signals and cytokines, for example IL-2, required for licensing of the dendritic cell. This licensed dendritic cell can then cross-present the exogenous antigen with an MHC class I molecule and activate a naive cytotoxic T cell. Besides activating naive cytotoxic T cells, this phenomenon of cross-presentation by dendritic cells can also induce tolerance against self-antigens. This occurs especially if the licensing by the helper T cell is incomplete due to lack of proper co-stimulatory signals and or appropriate cytokines, as happens frequently in case of self-antigens. In such situations, the naive T cells develop tolerance against that antigen. Till now, we have discussed the processing and presentation of peptide antigens. But what happens if the antigen is not a protein but a lipid or lipid-based antigen? The molecules which are responsible for presentation of these lipids or lipid-based antigens are called CD1 molecules. Like the MHC class I molecules, the CD1 molecules also consist of a heavy chain and a beta-2 microglobulin or B2M. The expression of these CD1 molecules is restricted to only the antigen-presenting cells or APCs and the double positive cortical thymocytes. In humans, there are 5 isoforms of CD1 which can be divided into 3 groups. Isoforms belonging to groups 1 and 2, that is CD1A, B, C and D, are capable of presenting lipid or lipid-based antigens. The group 3 isoform, however, which is CD1E by itself, is not capable of presenting lipid or lipid-based antigens. It is believed that it is a lipid chaperone and therefore indirectly helps in the loading of lipid or lipid-based antigens on the other CD1 isoforms. These isoforms of CD1 differ in their cellular expression, intracellular trafficking, and the kind of lipids they present. Now, the exact mechanism of antigen presentation is different for each isoform of CD1. But the most common pathway is, first of all, these antigen presenting cells which express CD1 molecules internalize the bacteria or aggregated lipids by either phagocytosis or endocytosis. The second step is the processing of these lipid-based antigens, and this involves deglycosylation or breakdown of these lipid aggregates or membrane structures into monomeric forms. And this is followed by their loading on the CD1 molecules. Here we need to remember that many lipid-based antigens do not undergo any kind of processing and therefore are straight away loaded onto the CD1 molecules. The antigen CD1 complex is then presented on the surface of the APC, and then it is recognized by specialized cells, for example NKT cells, which bear a cognate receptor. Since the CD1 molecules do not show any significant variation between different individuals, these molecules can act as excellent targets for vaccines. So students, let us now summarize what we have learned in this module. The first thing that we have learned is that a TCR can recognize its specific antigen only after it has been processed and presented with a self-MHC molecule. The other thing that we have learned is that the TCRs of CD4 positive and CD8 positive T cells recognize peptide antigens presented on MHC class 2 and class 1 molecules respectively. So CD4 presented with class 2 and CD8 presented with class 1 molecules. The third thing that we have learned is that MHC class 1 molecules are expressed by most nucleated cells. On the other hand, MHC class 2 molecules are expressed by only a few specialized cells which are known as antigen presenting cells, and their examples include dendritic cells or DCs as we call them, macrophages, B cells, and thymus epithelial cells. The next thing that we have learned is that the endogenous pathway leads to presentation of proteins in association with MHC class 1 molecule. The exogenous pathway, on the other hand, leads to presentation of proteins in association with MHC class 2 molecule. Now, besides that, we have also learned that proteasomes are the proteolytic machinery present in the cell which breaks down the endogenous proteins into peptides. And proteolysis of exogenous proteins occurs inside the M2C or late endosomal compartments, and this is usually brought about by proteases which are known as cathepsins. The next thing that we have learned is that chaperones and other associated proteins present in the rough endoplasmic reticulum help in proper folding and assembly of the MHC class 1 molecule. Another thing to note from this lecture is that the TAP protein mediates the transport of peptides from cytoplasm into the rough endoplasmic reticulum. The next thing that we've learned is that dendritic cells activate the naive cytotoxic T cells by a process known as cross-presentation, wherein exogenous antigens are presented on MHC class 1 molecule, which is usually involved in the presentation of endogenous antigens. And lastly, non-peptide antigens are presented with the help of CD1 molecules. With this, I would like to conclude this lecture. Thank you.